Welcome, everyone, to the ASTCT member webinar. This is your secretary, Christy Sullivan. We're happy to welcome Dr. Raymond Tice, Chief of the Biomolecular Screening Branch of the NIEHS, to talk with us today about the Tox21 program. Minor housekeeping issue, the webinar is recorded. A link will be sent to members after the presentation. Also, lines will be muted to cut down on background noise. After the presentation, I will unmute the lines in order so that you can ask questions. Finally, I wanted to remind everyone to check the website for resources and events, and let me know if you have any ideas for items that should go up on the website or ideas for future webinars. The web address is astctalks.org. So I'm going to go ahead. Ray, I think. Ray, did we lose you?
No, I guess I didn't know you wanted to talk yet. So you can mute and I can go ahead and tell you if you can see if you can still hear me.
Well, I don't see your number there anymore, which is rather strange. So I'm going to leave everyone unmuted because I can't.
There's not that many people. So
it's not too bad. Okay. All right. So go ahead. Thank you.
You've got one of one. So
Oops,
I
sorry.
guess you need.
There you go.
Okay, so we're a small group, so since we're unmuted, feel free to ask a question if you feel like it as we go through. And the purpose of this presentation is just to kind of provide an update of what Tox21 is and where we are in terms of our strategy of trying to replace, trying to move toxicology into a more mechanistic-based understanding of events that occur. So I'm going to go through and start clicking on slides, and you'll see it all on your screen also. So this is slide number 2. And basically one of the key documents that was released was by the National Academy of Sciences in 2007 called Toxicity Testing in the 21st Century, a Vision and a Strategy. This was there was a related document that was published a year or so earlier. This was the final report, and what I have on the right side of the screen is, let me get the pointer going, is just some information about, they say that they envision a not-so-distant future. We can talk about how long that future might take, in which virtually all routine toxicity testing will be conducted in vitro by evaluating perturbations in a suite of toxicity pathways using high-throughput robotic-assisted methodologies. What I found interesting was we were actually, in terms of NTP and EPA, already trying to do some of this stuff, but because government people are not on the panels, they actually didn't know what we were doing or how far we had progressed. But this document allowed us to point to it and say, this is the kinds of things that we want to be doing, and allowed us to put together a memorandum of understanding that was signed off at fairly high levels. The memorandum of understanding was actually released, I'm looking at slide 3, was released on Valentine's Day of 2008, and at the same day there was a paper that was published in Science on Transforming Environmental Health Protection by Francis Collins and George Gray and John Bucher. And you can see this is a diagram that comes from that publication, and basically it just points out kind of what throughput is. At the same time, it talks about the lowest throughput is on the human experience side, where there's immediate human relevance, but you can't do too many chemicals, and you're looking at something after the fact. And on the right-hand side, you're looking at where you could actually run, thousands and thousands of chemicals a day, but in high throughput, but you're much further away from immediate human relevance. And we recognize the importance of computational toxicology, the standard rodent toxicological tests, which we consider to be the legacy data, and the alternative animal models. And I'll bring some of those into play as we go through the talk. On the next slide, this is a slide that came from the National Academy of Science report, where they're talking about perturbations of a toxicity pathway. I think the name has now changed. They don't like to call it a toxicity pathway. The language that's being used at the OECD is to call it an AOP for an adverse outcome pathway. But basically, these are cellular pathways that normally get perturbed, but there's adaptive responses, homeostasis, that can take care of that, whatever that perturbation is. But so as you go up to higher and higher doses, you're going to end up in a situation where the perturbation is no longer can be taken care of by homeostasis or the adaptive stress responses, and you end up with cell injury and morbidity and mortality. And so this is considered the... Pardon?
Oh, sorry.
That's all right. This is the framework by which we are operating, is that we're focusing on pathways that we consider to be informative in terms of adverse health outcomes. On slide 5, it talks about the formation of what we call the Tox21 community. That was actually something introduced by Bob Kavlock. Basically, the original 5-year memorandum of understanding was on February 14th. It was signed off on by Francis Collins, Sam Wilson, who was the acting director of NIEHS at that time, and George Gray, representing the R&D side of EPA. And one of the reasons we're actually feeling supportive is that Dr. Collins, of course, is now the director of NIH, but he was very active in pushing this forward in 2008. So we feel that we still have support at the highest levels of NIH. And then in 2010, on July 19th, we released a second revised 5-year MOU that was now signed by Linda Birnbaum from NIEHS, Paul Anastas from EPA, Dr. Green from NHGRI, and Dr. Woodcock from FDA. So that was the time at when FDA joined this consortium. And we call this, or consider this, to be a community resource project. And a community resource project is a little bit like what it was what the Human Genome Project was. We recognize that what we're trying to do is to get data on all the different chemicals to which humans might be exposed that we often know little bit about, and our whole intent is to make this data publicly available, so at the same time while we're trying to develop our own prioritization schemes and prediction models so that the scientific community as a whole can use this resource in trying to understand the relationship between chemicals and toxicity. If we look at the next slide, on slide 6, that's the Tox21 screening timeline, and this just kind of puts things into perspective in terms of what was also happening at the EPA and at the NCGC at that time. So the NCGC was put together about mid-year on 2004. The EPA National Center for Computational Toxicology was initiated in the beginning of 2005. We published, there was an NTP vision and roadmap that was published also in mid-2004 that talked about using high-throughput screens. And in early 2000, we started interacting with the NCGC in 2005, but in 2006 is when testing actually started. And that testing was on a library of compounds that we provided, which was 1,408 compounds. And then about that time, a little bit later, EPA became involved, and they also sent up 1,408 compounds, coupled with a few more that they wanted looked at specifically. And that was the start of what we call phase 1 within Tox21. In addition, EPA in mid-2007 also started ToxCast, and ToxCast was a focus on broader biological space, but with a smaller number of chemicals. And so in that case, it was 309 unique chemicals tested across about 550 different assays. And then you can see that in 2010, we started phase 2 of the high throughput screening program at the NCGC with a 10,000 compound library, and I'll talk about that. And ToxCast phase 2 is 1,000 compounds tested across maybe about 600 different assays, and they're in the midst of that right now. If we look at the next slide, it kind of tells you the different areas of expertise the 4 partners have brought to the table. And at the same time, another reason for putting this up here, it kind of gives you an idea of the different areas that we consider to be critical for success within Tox21. So, of course, there's lab animal toxicology, there's human toxicology exposure assessments, there's ultra-high throughput screening, which is what we're doing at the NCGC. We have different kinds of low to mid throughput assays. We'll talk a little bit about that. There's stem cell assay development. We think we're going to have to move more and more into using stem cells as target cells in vitro. We've been looking at how to bring epigenetic assays into the mix. We think we're going to have to include engineered tissue models. We've been supporting the development of 3D tissue models in the liver and the lung and the skin. We've also started to try and integrate some of the other omics-based systems into what we're doing, whether it be in animals or in cells. We have lower organism systems. We have a C. elegans lab at NIH, and EPA does zebrafish embryo assays, and FDA does both of those. We've been looking at genetic variability and response with a focus mostly on human variability and response, and I'll mention that. And we've been developing databases, informatic tools, and we have some validation experience at different levels among the different partners. The goals of Tox21 are outlined here. Identify patterns of compound-induced biological response. The immediate goal really is to prioritize compounds for more extensive evaluation, and ultimately to develop predictive models for biological response in humans. One of the things I want to try and get across to people who, when I talk about this, is that there's been a continuing dialogue within the scientific and the public community and the regulatory community on whether or not in vitro assays can be used for making regulatory decisions. And one of the things that I hope will come out of the data that we're collecting and the comparisons we're making is that we will understand the extent to which that can be or cannot be. So whether we actually ultimately demonstrate that in vitro assays cannot be used to cover all different kinds of toxicities, that to me would still mean success, because at least then we would know what we could use it for and what we can't use it for, as opposed to just kind of arguing about it in the absence of real data. This shows the structure of the Tox21 community. The agency points of contact are listed above. Bob Kavlock was the point of contact for EPA until earlier this year, when he moved to a higher level position up in Washington, D.C. And David Dix, who was the deputy director for the National Center for Computational Tox, is now the acting director. You can see we're broken up into 4 working groups: assays and pathways, chemical selection, informatics, targeted testing. You can see who the co-chairs are. There's a representative from each of the 4 organizations. If you look down below and down here, you can see the different activities that they focus on. The first group was focusing on what are the assays that we should be having in our Tox21 efforts. The chemical selection, of course, focuses on what are the chemicals that should be in the 10K library, and I'll give you some information on that. we're actually then thinking about, after we've gotten that library in place, is trying to develop libraries of mixtures or aqueous soluble compounds that were not could not be put into the 10K library, which is DMSO soluble. We have an informatics working group. How do you handle the data? Develop prediction models, and we have what we call targeted testing, which is basically to try and see if there is a prioritization scheme or a prediction model that's been developed, are they actually relevant, and how do we test that? The other thing they're trying to do is to figure out how to extrapolate from in vitro concentration to in vivo dose. This particular chart right here shows you the kind of screening that we do at the NCGC. We often use either reporter gene assays. The one you're looking at, the chart on the right, this is actually a cytotoxicity assay. It's basically 1536 well plate format, 14-point concentration response curves. The chemicals we're using right now are DMSO soluble. The typical concentration range is 5 nanomolar to 92 micromolar. Assay volumes 5 microliters, and it's typically 1,000 to 2,000 cells per well. This is just a series of curves for a compound in a range of chicken cell lines that have been knocked out for different DNA repair pathways, but showing a little bit of the nature of the response. Obviously, this is a good response in the sense that all of us looking at it would say, oh, this compound is cytotoxic, and these cell lines over on the left show more sensitivity than the wild type, which is this blue line right here. So clearly not all the things are that straightforward, and I'll get back to that in one of the other slides. This is the screening throughput and why we've gone to 1536 well plate. This is the same footprint size as a 96 well plate, but you can see that the wells are much smaller because there's so many more of them, and that is another reason why we've moved over to robotics or automated systems for handling things like this. But this little chart down below gives you an idea about what the throughput is if you assume that you can handle 100 plates per day. And this is why we've moved into high throughput screening, which in fact does have its limitations, and I'll mention those a little bit later. This is a plate design for an assay that was used for cytotoxicity. We've got negative and positive controls over here. The reason our library was 1,408 wells or substances is because that's the number of wells that are available in a 1536 well plate once you have this plate design. And what we basically do is each plate is a separate concentration, and each plate gets normalized against the negative, and the data on it gets normalized against the negative and positive control data. And what you end up with is a stacked number of plates, in this case 14 plates, covering the 14 different concentrations. This next slide actually gives you a little bit of what it looks like. This is a Jurkat cell cytotoxicity assay. The compound is, there's 1,408 compounds on these plates. If you look at this well right here, this is a compound that shows activity at 0.003 micromolar. And you can see that if you follow this well across the plate, it actually shows up on every plate. And so basically you're going through and across the range of concentrations, finding the concentration response curve for each well, and then using that to make a conclusion about the particular compound that's in that well. So what we call phase one was directed at proof of principle. As I mentioned, we have provided 1,408 chemicals, 1,353 unique, 55 duplicates. The EPA also provided, and they ended up with 1,462 chemicals, 1,384 unique. And basically, we ran across about 100 different quantitative high-throughput screens. In addition, EPA was screening its 320 compounds, 309 unique, primarily pesticide active, some endocrine active compounds, and about 550 assays. And the data have been, are still being released as we go through and make sure we don't got any problems with the particular data set we have. Through EPA Actor, there's a link there, the National Library of Medicine PubChem, and also through a database at NTP called SEBS, and those are the links to get to them. ToxCast Phase 1 in April 2007. These over here, the list are contractors, and you can see the name of the contract: NovaScreen, Adagene, ASEA. These are the different assays that were run in those particular contracts. They also then added some in-house work on neurite outgrowth, cell proliferation, zebrafish. And then over here on the right on what they call ToxCast 1.2 was where some of the other CROs out in the scientific community under material transfer agreements agreed to test the compounds for free as long as the compounds provided, in the hope that their data would be integrated back into all of the data being developed. And down here you can see where we also screen those compounds in C. elegans using a developmental assay. In phase 1 at QHTS, the quantitative high throughput screening done at the NCGC, we have a series of assays. These are called phenotypic readouts: cytotoxicity, caspase, apoptosis, membrane integrity, mitochondrial tox, a bunch of assays that we relate to genotoxicity, cell signaling. There's a single epigenetics assay. These are biochemical assays, the ones on drug metabolism for some of the major CYPs. Target-specific assays, these focus on nuclear receptors, hERG channel, and then a bunch of different targets. One of the things that the NCGC was doing is because they were part of the Molecular Libraries Initiative, and the purpose of that was to screen low molecular weight compounds for biological activity in different targets. Whenever they would run one of those assays, they included the NTP 1408. So we ended up with a bunch of different assays that are probably not very, some of them are probably not very useful from a toxicological standpoint, like anthrax lethal factor, but we do have some data on some interesting assays. And then one of the things we also did is we ran 87 HapMap cell lines. These are lymphoblastoid lines that have been densely genotyped. This is the Ceph panel, which is a panel of trios from Utah. This was a collaboration with Ivan Rusin and his staff at University of North Carolina. But the purpose of this was to start having a conversation about, could we use the same approach that we're using in high throughput screening to look at something about the genetics of variability and responsiveness among cell lines, where we could actually look and see what the DNA sequence was. This slide shows the first thing that got published and got evaluated was to go through and compare the toxicity of the 1,408 compounds against 13 cell types. There are 9 human, that's H. There are 2 mouse, M, and 2 rat, R. This is a heat map that shows the relationship between the cell lines and the compounds. And one of the things, and the reason these cell lines were picked were they represented different organs. Although some of these were cancer cells, some were transformed, and there's a few primary cell types in there. And you can see across these 13 cell types, there are some compounds that actually are toxic, cytotoxic to all the cell lines. These tended actually to be compounds that contain mercury. And then you can see, like up here, here's a set of compounds that only hit N2A cells but don't hit any other cell type. So one of the things we're still trying to figure out is what's the best cell type to use in a screening approach, and kind of what we're focusing on. If we were looking at cytotoxicity only, we'd probably go to the cell type that had the greatest number of hits, which tended to be JERKID or NIH3T3 cell. That would be the one that would have the greatest sensitivity. But we still recognize there are some unique compounds that even wouldn't show up in those cell lines. In the next slide, what I've got is not a heat map, but this is just based on all the assays that were run. To get kind of everything on a page, we've tried to reduce multiple assays that were similar in nature, like all the cytotoxicity assays were basically reduced to 1. But what we have here is stress response pathways, some nuclear receptors. Some CYPs are indicated on the bottom. Here are the P450 CYPs. And you can see if you look at hit rates, these particular assays had more hits than all the other assays in and of itself. This just gives you an idea about the overall hit rate as we got between the different assays that were run. In phase 2, ToxCast phase 2 is about 1,000 compounds being screened across about 600 different assays. If you look at what we're doing up at the NCGC, we have what we're calling a 10K library, and I'll provide some more information about that. But we're actually screening it 3 times in each assay. One of the things we ran into is that when you look at the data, there are compounds that are clearly active in a particular assay, and there are compounds that are clearly inactive in that same assay over the concentration range tested. But there are also compounds that we classify as inconclusive, where it looks like there's some activity, but it's not enough to make you feel comfortable with claiming it as an active compound, but there's too much activity to call it negative. And so what we decided to do is, at least initially, is to go through and test the 10K library 3 times in each assay, so that we would generate at least 3 concentration response curves on each substance. And that way we might be able to move some of the compounds that are classified as inconclusive into one category or the other. And at the same time, it would give us a better estimate of what the AC50 is for a positive chemical, which might be useful in looking at potency. So that's why we're doing it 3 times. And we decided to focus initially on nuclear receptors, both activation and inhibition. You can see the range of nuclear receptor assays here. One of the things we further decided to do, because we had a lot of discussion, especially about ER and AR, the estrogen receptor and the androgen receptor, the assays that were used in phase 1 use partial receptors, basically the binding domain and the C-terminal in, which is what's primarily used in pharma. But we also had researchers who were working with the full receptor who believed that there were some compounds that did not act by binding, but did modify the receptor and did stimulate estrogen receptor activation. So we decided to actually put both kinds of receptors in. We have both a partial and a full receptor for ER, and we're trying to put in a full receptor for AR that would work in high throughput screening. I might mention at this time that not every assay is amenable to use in 1536-well format using robotics. And the reason for that is that the assays have to be homogeneous. And what that means is that you can add things to the wells, but there can be no rinsing step, or you can't take things out. They don't have it's not something that can be done considering the small volumes that are used. And furthermore, the assay can't last longer than about 48 hours. Otherwise, they run into problems with loss of volume. So basically, we can't do everything every assay is not amenable, and also the signal to noise ratio needs to be at least 3, but preferably higher. So, and then the other set of assays that we're running are ones that relate to stress response. These include DNA damage, heat shock, hypoxia, oxidative damage, inflammation. That's the other set that we're running. And as I think it says here, we're looking at nuclear receptors both for activation and also for inhibition, agonism and antagonism. So this is how we decided on the assays, and I want to mention that as we're running through this particular library or this particular battery of assays, we're starting to think about the future. And in the future, we're trying to figure out how to bring in disease-associated pathway, a focus on pathways that might relate to like obesity or diabetes or autism. Those are things that we have an interest in. But using stem cells either in an undifferentiated or a differentiated stage. And we're trying to move into more high-throughput gene array assays. And those are some of the things that we actually have pilot studies ongoing right now. If we look at this one, just as a picture of Shortly after the March SOT meeting in 2011, we had a ribbon cutting that inaugurated a new dedicated robotic system. The one that we typically used in the past had like 3 of these arms right here, but there's a single arm. And if you're interested in watching this thing being put together in a short video using time-lapse photography, here's a YouTube thing that's kind of interesting to watch. But this is the design of what we're using in high throughput. As far as the compound library, the 10K library, we started, and this was it was mostly driven by people at EPA. They put together a list of all the compounds anybody could think of that either were in somebody's regulatory oversight, was a concern for different organizations, and we're talking including outside the U.S. And so we ended up with a list of about 120,000 compounds. Once you remove duplicates and you only want to focus on things that have uniqueness, not a mixture, you ended up with about 19,000. There were about 15,000 with chemical structures, which is what we wanted to do. We ended up with about 11,000 compounds that theoretically could go into a library. And then over here on the left are some of the things that we needed to have, like we needed to have a molecular weight range in this range of 100 to 1,000 molecular weight. And we wanted to make sure that they were soluble in DMSO at some reasonable level and that they were not likely to be gases and therefore disappear from the plates. So once we had that, we ended up with a 10K library or so that has NCGC's component was they had put together a drug library, drugs, drug-like compounds, active pharmaceutical ingredients. EPA's library included both the ToxCast Phase 1 and Phase 2 compounds, endocrine disruptor screening program, the FDA drug-induced liver injury project, but they were fortunate enough to also get from different pharmaceutical companies about 135 drugs that failed in clinical trials. And so this is really a major advance because the pharmaceutical companies not only gave the failed compounds, but they're also willing to provide the structures and the data associated with why they failed, and they agreed to make this data public. And that's, I think, a unique event in the history of what we're trying to do. Over on NTP, you can validation reference compounds from things that they had been gathering for regulatory evaluation. And one of the things that we put in was a set of formulated mixtures. These are mixtures that include, there's a mixture for ER, there's about 10 compounds. In various both binary, tertiary, and up to 10, there's another mixture of AR compounds. And then the one that's the most interesting, for me at least, is there's a mixture of compounds that are cytotoxic. There are 62 individual compounds in that mixture in various proportions. And so we have both the individual compound, each individual ingredient, constituent, and then we have them in various sets of mixtures to give us an idea whether or not the kinds of responses we see will be additive, antagonistic, or potentially synergistic. This gives you the compound library. This table down here tells you how many compounds were provided by each of the 3 partners. There's actually 8,193 unique compounds. There's a total of 11,776 compounds in wells. Some of the compounds are duplicates. This chart over here on the right gives you an idea of how many duplicates there are. These are not planned duplicates in the sense that we went out and got them from the same source, but these are duplicates because we had, for instance, EPA wanted to have compounds in their ToxCast libraries, and we wanted to have compounds that were being tested at NTP, so therefore there's a reasonable amount of overlap, and we're looking at this as a way of evaluating replication within the assay. In addition, we have 88 sole source duplicates on each of the plates, each of the 1536 well plates, so that we can look at within plate and across plate variability for a set of compounds that were selected because based on phase 1 data, they tended to have some number of hits in each, in many of the different assays that we had. The Genomes Project with Ivan Rusin at UNC, I mentioned in phase 1 we did 87 cell lines. However, in phase 2, we moved that up to 1,090 cell lines, representing 9 different racial groups. And against those cell lines, we tested 180 compounds selected because in previous studies they had shown some level of cytotoxicity at 8 concentrations, measuring a cytotoxic response. And currently they are evaluating the data to look and see for those compounds for which there's lots of heterogeneity in response across the 1,090 cell lines. Can you link that differential response to differences in genes? There is a presentation tomorrow, which is actually webcast, from the National Academy of Science, and I can look for it after I get off of this if anybody's interested. And there will be a presentation on, the first presentation on how this data is coming out in terms of identifying different genes. And there was a poster on it at SOT, by the way. But for us, it's a way to start thinking about using high-throughput screening not only for identifying compounds, but for looking at the extent to which different individuals with an underlying differences in genetic background have different sensitivities or susceptibilities to different cytotoxic or toxic compounds. One of the things that we've done recently, which is not high-throughput screening, is last September we acquired something called Drug Matrix, which is a rat toxicogenomics database. It had been put together on 637 compounds. The little chart, the circle chart on the right, pie chart, gives you some idea of what the compounds are. Most of them are drugs that were approved, but there's also other chemicals in here. And one of the went through and did is they did drug treatment transcript profiling, they did histopathology, they did blood chemistry, they reviewed the literature, and they came up with a set of comprehensive signatures for the difference between chemicals that are liver toxicants and ones that are not liver toxicants. They were selling this information to pharmaceutical companies. And then when it moved over to another company, they decided that was not within what they wanted to do. So we managed to acquire it, and all of this data is now available through these websites down here. There's the Drug Matrix website where you can actually upload your own genomics data and see how it compares to other ones. And then there's something called ToxFX, which is a way of getting a report out in a very short time and without very much training. So we think it's extremely useful as we start moving into gathering more data on how genomic signatures might help us understand whether a chemical might be a potential toxicant of some kind. This slide here just tells you the number of chemicals for different tissues. When Iconics ran these assays, they focused on chemicals that had adverse effects or not adverse effects at a particular tissue like liver. So liver was the biggest one, but you can see they have gene expression, chemistry, histo, pharmacology literature, and in fact they also did some primary rat hepatocyte work too. One of the other things that has been developed at the NCGC, and hopefully this will be released in the near future, is they put together a universe of human pathways. There are 1,100 human pathways that have been mapped to this pathway globe. The globe actually rotates. If you look on up here in the corner, you can actually go through and select toxicity pathways or disease pathways that have been identified in the literature. You can put in your specific gene. You can look at the relationship between any two pathways, like between P53 and hypoxia-inducible factor. Where do these interrelate? And so one of the reasons that this was put together was so we could get some idea of how much coverage we have across the human pathway map. And so right now we're only covering about 30% of all the possible pathways. But one of the things we also wanted to do was to look and see how many nodes there were in these 1,100 pathways, and it turns out there's about 250 nodes where pathways intersect. Some of the nodes are not very useful because they cover more than 100 pathways, but by having this knowledge and seeing where our assays inform us, we can start looking toward being able to cover the complete human pathway map. The other thing we're doing is trying to figure out how to mine the NTP archives for disease gene signature. And so if you look on down here in the lower left, you can see that the number of studies, how many histology blocks, there's 54,000 frozen specimens, lots of things in paraffin. So what we've been focusing on, since we already know that frozen specimens can be used for most of the omics analysis we're interested in, we've actually been running studies on formalin-fixed paraffin-embedded tissues to see if we can get out DNA for epigenetics and RNA, looking at some different platforms that might allow us to go through and go back into archives for for samples that have been taken from animals that have been treated with specific chemicals, and see if we can identify the gene signatures that are associated with a particular toxicity. So one of the other things we've been trying to do is work with some partners to develop databases that could be used. I mentioned SEBS, but we've also worked with LeadScope through a joint agreement with LASA and Multicase to develop what LeadScope is calling an integrated prediction system. And basically it's a system to help us prioritize chemicals through human relevant toxicity predictions. It'll have the data from Tox21, the historical in vitro and vivo data, and the QSAR models for adverse events. And so this particular thing would be by subscription, but we're trying to get something together that would make life a little bit easier for people as they start looking at what different chemicals might be capable of doing. So what are the critical high-throughput screening issues? One is that cells don't get diseased. Not all compounds can be screened. The biggest limitation we have right now is we don't have xenobiotic metabolism incorporated in 90% or more of the in vitro assays. We cannot use S9 mix like typically used in gene tox assays because you'd have to wash it out to be able to do it. We need to consider interactions between different cell types. That gets back to the 3D models. All of the exposures we're doing acute, but what about chronic exposure? We need to keep on measuring human variability and see what that can tell us. We need to extrapolate from in vitro concentration to in vivo dose. There's a number of people, scientists working in that arena. We need to identify disease-associated pathways and useful assays. In that case, a year ago, January, there was a meeting on diabetes and obesity, a workshop sponsored by NIEHS. And one of the things is it brought, excuse me, it brought together epidemiologists along with informaticists and people who were working in basic research on those diseases to try and identify pathways and useful assays, and we're trying to move forward on that. And then ultimately we need to integrate multiple data sources. So good thing this is the last slide. My voice is going. So these are the bullets I've written that say what success depends on. One of the things I didn't mention with regard to the chemical library, the 10,000 chemical library, is that we are doing quality control to determine the identity, purity, to the extent we can, the concentration and the stability of all the compounds that are in that library. And that in itself is not a trivial exercise. When you think about a lot of these things are not amenable to easy chemical analysis. We need to characterize the assays in terms of reliability and relevance. That's one of the things that we always try and do. We have to be able to incorporate xenobiotic metabolism. We need the informatic tools. We need to understand the relationships between chemicals and genes and pathways and disease. We need to make this data freely accessible for anybody who might be interested in working with it. And we need to both have scientific outreach and training of the next generation of scientists who would be, and regulatory scientists who would be using the data that's generated by the system. And that's the end of what I have to say, but I would welcome any questions anybody might have.
Well, thank you very much, Ray, for that presentation. A lot of new stuff going on. Does anyone have any questions? I have a question about the human pathways map that you talked about.
Mm-hmm.
You said that it was going to be made available. What is it going to be on the web or published in a publication?
No, they have a draft paper, but that they want to get out to describe what this does. But it'll be available on the web. It'll be available through a location on the NCGC website, and they're no longer called NCGC. They're now called NCTT because they moved over from NHGRI to another acronym to NCATS. So they've gone through a name change, but at some point in time, there will be this thing available, and what I plan to do is as soon as I know it's available, I will pass it on to you, Christy, so that you can distribute to the members of the organization.
Sure. What I would like to know is it going to be something that people can add to and that it will grow, or is it going to be a static information resource?
Well, it's going to grow as we get more information about the relationship between genes and pathways, but I don't think that it would be amenable to growth from the outside. I think what we're going to do is just do updates with it periodically based on where the information came from in the first place. I don't know that it'd be something that anybody could say, well, here's another pathway, and how do we put it in? And that question has never come up before, but I will ask them.
I know that someone else is working on a pathways wiki that people can add to from the inside or the outside. Mm-hmm. So I wasn't sure if it was a similar effort.
I think one of the issues is that the reason it's really a work in progress is that pathways change as more information comes in. Mm-hmm. And so, and new pathways get identified and new relationships between pathways get identified. So and I don't know if there is, it's just like any other data set, well, you might be able to add something for your own use, but I don't know that it would become commonly available unless it went through some kind of quality control step.
Right.
One of the things we have to worry about with the internet and the ability to get information is not all information is equally valid.
Sure, definitely a concern. Does anyone else have a question?
I have a question. This is Craig Rowlands from Dow Chemical.
Hi, Craig.
Hi, Ray. Thank you. That was a really nice overview of what is obviously a tremendous amount of work going on in different areas. One of the things that I'm curious about, and it's a challenge, why we probably don't see a lot of this, is the ability to extrapolate some of the exposures, concentrations that are observed in these in vitro assays to what the human equivalent exposure would be, and what efforts might be going on there to try and keep up the pace of that exposure assessment to the screening assessments.
So there's different activities, but I would say the biggest place where this is occurring is at EPA's side. So they have, in addition to ToxCast, they have what they call ExpoCast. And if you actually Googled EPA NCCT, National Center for Computational Tox, and if you look at ToxCast even, on the left-hand side there's a list of different activities, and ExpoCast is one of those. And so they've actually been trying to bring some exposure data into the prioritization scheme for endocrine disruptors, for example. Try and get an idea if all we do is prioritize based on activity, we might want to take a compound that had a little bit less activity but a lot more human exposure as in terms of the prioritization. In terms of the actual levels, there has been some work trying to relate what's picked up in NHANES versus what's going on within whatever the AC50s are for a set of compounds that are active in a particular set of assays. But I know that, so there's a group at EPA within the ToxCast group that are doing that, and I also know at the Hamner, Rusty Thomas has been looking at trying to do what they call reverse toxicokinetics to go from an effective concentration in vitro to an effective dose in vivo. At the same time, we have some work within NTP. Mike DeVito's been looking at a couple different data sets where there's both in vivo data and in vitro data to try and see how the active concentration correlates between those two arenas. But I will admit that it's extremely difficult because you have to kind of take a lot of assumptions into place when you do that. An animal or a human is a kinetic process, and a tissue culture flask is fairly static. So that's one of the areas where it's going to take a lot of work.
Okay. Thank you. That was
sure.
Hey, Ray, this is Judy Strickland. There was an article published with Barbara Wetmore as the senior author not too long ago that did the in vitro to in vivo extrapolation, and my recollection is they also did a population exposure evaluation as well. I think that was published in 2011, 2012. I don't remember the journal.
Well, it's actually, if it's out of that group, then if on the right-hand side of the EPA website for NCCT, there's a list of there's a publication icon, and it lists all the publications. So somebody could easily find the publication there. And I've seen enough slides that I can remember one where they were talking about a set of compounds tested in the 309 ToxCast phase 1 with regard to human exposure levels, and therefore based on the activity AC50 of a particular compound, whether it even fell within a range of concentrations that people might be exposed to. I've got that figure in my head, but it probably is in the Westmoreland paper. Any other questions? No, then thanks very much. My email address is on that first slide. So if there are some questions or queries, somebody would like more information, we think the more people that know about and understand what we're trying to do and make suggestions or provide critical comments, the more chance for success we'll have in actually figuring out to what extent this particular approach is useful or not useful. But thanks for listening.
Thank you, Ray. Thanks for the presentation. And this concludes the webinar.